Hi, I'm Matti Lehtinen from Karolinska Institute, and I'm going to talk about vaccine-induced immune responses, the sustainability, and HPV vaccine efficacy. I was collaborating with Cambridge tumor virus group already 30 years ago when I received from Professor Lionel Crawford virus-like particles expressed from the fission yeast Schizosaccharomyces pombe. And they were one of the first set of virus-like particles in this field used in serological assays for the detection of HPV type 6 and 16 antibodies in naturally infected individuals. Later on, the vaccine companies used yeast and baculovirus expression vectors to generate virus-like particles that ended in VLP vaccines. And the general understanding is that seroconversion following vaccination is 100%, and that's it. I'm not sure if we should accept this 100%, and I will go in detail into evidence that this may not be the whole truth. However, some background first. We know that the vaccine-induced antibody responses are the better the younger are the vaccine recipients, and this is evident from our studies in early adolescent and young adults receiving the bivalent vaccine, where we could show that the antibody response to the bivalent vaccine was statistically significantly higher in the early adolescents 10 to 14 years of age as compared to young adults. There is also a difference in between the two vaccines, bivalent and quadrivalent vaccine. The bivalent vaccine induced antibodies are more cross-reactive to types especially in clade A9, types related to HPV-16, that is 31, 33, 52, 58, and also the antibody titers are way higher in a logarithmic scale as compared to the quadrivalent vaccine-induced antibody titers. We also know that the antibody levels induced by the quadrivalent vaccine wane over time, especially those conveyed by HPV-18 component of the quadrivalent vaccine. An interesting observation we made in the vaccination studies with the bivalent vaccine was that individuals who had no cervical antibodies against HPV-16 or HPV-18 had significantly lower serum antibody levels both to HPV-16 and HPV-18 as compared to those who had cervical antibodies against these HPV types following vaccination. We will come back to this observation in terms of possibilities to identify protective antibody levels. It is a reasonable assumption that the serum antibody level has to be at the level where cervical antibodies are detectable. To sort of summarize the background observations that we have is that the vaccine-induced antibody levels are highest in children. Bivalent vaccine is more cross-reactive than quadrivalent vaccine. The HPV VLP vaccination induces 100 times higher higher antibody levels than natural infections, and the HPV-18 VLP antibodies may not be sustainable for 5 years in the quadrivalent vaccine recipients. And it is highly likely that low levels of serum antibody following vaccination is associated with lack of detectable cervical antibodies. We had a possibility to look for the long-term effect of vaccination both in the quadrivalent and bivalent vaccine trial cohorts that were enrolled in Finland first to the Future 2 study in 2002 and 2 years later to the Patricia study. Sizable cohorts of 16, 17-year-old girls were participating in the trials. What was important in the Finnish context was that we consented the trial participants to registry-based follow-up because Finland is a wonderland of health registers maintained by the government, and the social security number is a mandatory feature of Finnish individuals. You don't exist in Finland without a personal identification number. So we could use this personal identification number that we had obtained via informed consent from the vaccinated individuals to link the vaccine trial registry to the Finnish maternity cohort shown in yellow here. The Finnish maternity cohort is a large serum bank established in 1983 and contains first trimester serum samples from virtually all pregnant Finnish women over more than 30 years that have been collected by the Institute for Health and Welfare until 2016. And there are altogether 2 million serum samples from 1 million women. We used this knowledge and the information that approximately half of the Finnish women become pregnant by age of 30 for the long-term follow-up of vaccine-induced antibody responses in these two trials. Patricia trial with the bivalent vaccine on the left-hand side, shown in orange, and the Future 2 trial shown in green on the right-hand side. We could identify sizable numbers of females who had donated serum samples to the Finnish maternity cohort up to 12 years post-vaccination, and we were retrieving these serum samples and detecting vaccine-induced antibody levels by the VLP ELISA method. Antibodies binding to the VLP or actually pseudovariants of different HPV types 6, 11, 16, 18, but also the cross-reactive types were available. And we were also using a neutralization assay for a number of HPV types 6, 11, 16, 18, 31, 33, 30, 45, 52, 58, which was of course important as we will see down the road. The first observation that we made was vaccine-induced HPV-16 and HPV-18 antibody levels over time by 12 years post-vaccination. And the important observation was that the bivalent vaccine-induced HPV-16 and HPV-18 antibody levels were significantly higher than the quadrivalent vaccine-induced corresponding antibody levels. This was true also for the neutralizing antibody levels for HPV-16 and HPV-18, and it is remarkable how big the difference between HPV-18 antibody levels induced by the bivalent vaccine and quadrivalent vaccine is. It is even comparable to the very low cross-reactive antibody levels to HPV-6 in bivalent vaccine recipients, lacking actually the HPV-6 component from the vaccine. What was sort of interesting and repeated over time with different methods that we used for antibody analysis was that the HPV-18 vaccine component in the quadrivalent vaccine recipients was not inducing antibodies in 15% of vaccine recipients, shown in green here, whereas the orange bar is showing 100% seropositivity for HPV-18, HPV-16 antibodies in the bivalent vaccine recipients. The antibody level was significantly different, both as for HPV-16 and for HPV-18, but this is a characteristic feature known already in earlier studies. What was new was when we were comparing the nonavalent Gardasil 9 vaccine recipients to the bivalent vaccine recipients in a head-to-head comparison, and the observation was similar. The antibody levels in the bivalent vaccine recipients for HPV-18, but also for HPV-16 antibodies were significantly higher in the bivalent vaccine recipients. This was true to some extent also to HPV antibody avidity that we were measuring, and especially so for HPV-16-18 components of the bivalent and quadrivalent vaccine. And this is important because antibody avidity is a measure of the availability of T cell help, without which the maturation and maturation of antibody avidity does not really happen. So one might wonder whether this has to do with the HPV-18 antibody response, and especially whether the adjuvant in the bivalent vaccine is superior in generating the T helper cell response responsible for the higher avidity. The other observations that the VLP-induced antibody levels, when detectable, they were generally sustainable, and that the bivalent vaccine induced antibody levels were much higher than in the quadrivalent or nonavalent vaccine recipients were also observed. What we did was that we continued the head-to-head comparison by comparing the antibody responses on an individual basis by the measurement of total VLP antibodies and neutralizing VLP antibodies, and what we could observe was that the antibody responses were significantly higher as for HPV-18, but also that the antibody response in 15% of the quadrivalent vaccine recipient was lacking. This is far-fetched comparison, but what we know from available data as for vaccine efficacy against invasive cancer is that the vaccine efficacy of the quadrivalent vaccine against invasive cancer is 88%, and vaccine efficacy against the bivalent the bivalent vaccine efficacy against cervical cancer is 100%. The 12% difference is reminiscent of the lack of HPV-18 antibodies in the quadrivalent vaccine recipients. Whether this will ever materialize as a difference between HPV-18 associated invasive cancers in the quadrivalent versus bivalent vaccine recipients is something that warrants surveillance. What we know so far is that actually up to 17 years of registry-based follow-up based on the countrywide Finnish cancer registry is showing highly similar vaccine efficacy against high-grade SIL plus, including invasive cancers, 65% vaccine efficacy for the bivalent vaccine in intention to treat analysis, and 68% vaccine efficacy up to 17 years in the quadrivalent vaccine recipients. This is actually identical vaccine efficacy and reassuring as for the use of both quadrivalent and bivalent vaccines in the vaccination programs. What was interesting was that we observed a correlation between cross-reactive antibody response seropositivity to clade A9 HPV types and vaccine efficacy against these same types in the bivalent vaccine recipients, whereas for the quadrivalent vaccine recipients, this was not observed. Taken overall, the differences in the bivalent and quadrivalent vaccine responses may be due to the folding and availability of the antigenic sites in the VLPs due to differences in the expression vectors. They also may affect the glycosylation of the antigenic epitopes, which is known to be different in the yeast-derived versus baculovirus mammalian, insect cell-derived antigens. And through, for instance, in the hepatitis B vaccine context. So some reflections of the observations. HPV antibodies are not induced by the yeast-derived VLPs in 15% of quadrivalent vaccine recipients, at least at the detectable level. Vaccine efficacies of the different vaccines against invasive cancer may be different. This is a study that is requiring continued surveillance, especially because the vaccine efficacies against high-grade SIL are similar. Bivalent vaccine efficacy against persistent infection with clade A9 HPV types correlates with neutralizing antibody response against corresponding types, which is sort of reassuring on the biological relevance of this cross-reactivity following vaccination with the bivalent vaccine. And availability of B cell epitopes may differ by the expression vector used. One black box in the antibody studies following vaccination with HPV vaccines is definition of protective antibody level. And to approach this, we have been following up the birth cohorts 92 to 95 that were vaccinated in a community randomized trial between years 2007 and 2009. The follow-up has been materializing in a randomized trial on different screening modalities in vaccinated women and has included visits at the age 22, 25, 28 with cytological HPV DNA and blood samples on each visit. We have so far identified in these vaccinated women breakthrough high-grade SIL cases, 44 breakthrough cases, and we have been able to type in an interim analysis the first 17 cases identified. The half of the high-grade SIL cases are showing a persistent infection with non-vaccine types like 33, 35, 51, 52, 59. And the important thing is that these persistent infections have materialized as high-grade SIL lesion in these HPV 16-18 vaccinated women. it was sort of surprising that the methylation status of cervical cancer risk genes was mostly negative in these women, positive only for HPV-33 associated high-grade SIL cases. And studies for the use of methylation markers in these in the triage of these high-grade SIL lesions in vaccinated women is ongoing. What is also ongoing is a kind of a early stage triage using these high-grade SIL breakthrough cases and their serum samples for the evaluation of a possible protective antibody level that would be detectable and guaranteeing that above this antibody level no high-grade SIL breakthrough cases are detected. This is the power calculation showing that already the first 17 cases that we have identified gives ample power with a cumulative high-grade SIL incidence of 4.5 in these women that are attending the screening trial. We have identified up to 44 cases by this time. The serum samples have been analyzed for pseudovirion ELISA, the binding VLP binding antibodies at Karolinska Institute, and the neutralization antibodies at DKFZ. And we are comparing the antibody titer distributions in the high-grade SIL cases and in the vaccinated and unvaccinated cohorts to identify in a stratified analysis the protective antibody level guaranteeing at least 90% protection. Whether this is doable or not depends on the biology of vaccine-induced antibody response. There's a high correlation between the 16 and 18 vaccine-induced antibodies in these vaccine recipients, but not so much for HPV-16 and HPV-33, the latter being cross-reactive antibodies, and there is a possibility to approach level of antibodies that may or may not protect against cervical high-grade SIL lesion. Whether this is through the presence of cervical antibodies in women with a protective level of serum antibodies is under a study. So a few reflections of the observations. 7,000 women are attending the surveillance, have been attending now for 16 years. And we have ample power for the protective antibody level early stage triage studies, but also for the methylation marker late stage triage studies with regard to the breakthrough high-grade SIL lesions. There is an analogy with protective hepatitis B antibody levels in a number of points, one of which is that the expression vector may have an effect on the availability of the important antigenic determinants, major epitopes of the VLPs. We are confident that vaccine-induced antibody levels that protect against high-grade SIL will be determined. Thank you.